Okay, on this lecture, I'm going to talk about the cell membrane and movement of substances across the cell membrane. I'm not going to go into a lot of detail, but hopefully some of the things I'm going to tell you will help. So if we look at a cross-section of the cell membrane, then you have the outside of the cell, the inside of the cell. Extracellular fluid on the outside, intracellular fluid on the inside of the cell. Substances have to pass back and forth, either waste material out of the cell or nutrients into the cell, for instance. And substances pass through the cell membrane in various manners. So, for instance, some things like water, oxygen, carbon dioxide move between the phospholipid molecules very quickly. Other substances need to use the proteins as channels. For instance, this one is a channel protein, which has a tube down the center, and material can move back and forth across it. Or carrier proteins, where the substance binds on one side and is flipped across into the other side. So these are the different kinds of proteins. I'm not going to go into all of them, but the different proteins have different types of methods for getting materials across the cell membrane. And again, here's an example. Passive transport versus active transport. In passive transport, materials move back and forth across the cell membrane without the use of energy, energy being ATP. If we have to do active transport, we're going to have to use ATP in order to get those substances across. Okay, over here, I've set up a flow chart to try to give you an idea of how those things take place. So, transport across the cell membrane. It takes place in two ways, like I said, either passive, no energy, or active, requiring energy. Let's follow the passive part first. So in passive, you have diffusion, which is materials moving from an area of high concentration to an area of low concentration. You can have simple diffusion, which things like water, oxygen, carbon dioxide can do when levels are not high enough. For instance, if you don't have enough oxygen in a cell, then oxygen can easily diffuse into the cell where needed. And you can have facilitated diffusion. Facilitated diffusion just uses one of the channel proteins, like I showed you, or a carrier protein, where it can bind to the substance and flip it in without using energy. If we have to use energy, we call that active transport. Active transport is going to utilize a variety of different methods. You can have primary transport. Primary transport uses a pump protein. So, for instance, the NAK pump that you've learned about is one of the pump proteins that's going to move sodium back and forth and potassium back and forth across the cell membrane. Secondary transport, or co-transport, then, uses the form of primary transport, but what it does is piggyback on top of it. So as the sodium's going into the cell, for instance, certain substances can hold on and get brought through along with that substance. If they go in the same direction of the substance, for instance, if they're going with the sodium into the cell, we call that symport. Sym in Latin means the same. If they go in opposite directions, for instance, as sodium's going in, an amino acid maybe can go out, we call that antiport, where they're going in the opposite direction. The other form of active transport, which requires energy, is called vesicle transport. And we didn't have enough room, so we'll go over here. There are two forms of vesicle transport: endocytosis and exocytosis. Endocytosis is the cell bringing substances into the cell using vesicles. So pinocytosis and phagocytosis are the two manners in bringing material into the cell in large quantities. So for instance, with pinocytosis, it's going to be water and nutrients, let's say minerals and vitamins. With phagocytosis, if you think about an amoeba has big arms and it wants to eat a bacteria. It's going to engulf that whole bacteria and take it into the cell. The way it does that is with phagocytosis, where where the cell membrane keeps growing around and around the bacteria until eventually it fuses. Now that bacterium is encased in a little vesicle. With exocytosis, now it's the opposite. We're going to get rid of a large amount of substance from the cell. So we use a vesicle. The best example of this would be tears. Tears are salty water. So as we cry and we're sad, we utilize vesicles in the cells. The vesicles are like a bubble, and the bubble encases around the water and the salt, goes to the end of the cell membrane, and then that gets exuded out of the membrane, or exo meaning outside.